I can outline the concept here while Allison is starting this. And, oh good. So we've had the good fortune to have access to some fresh human embryos, which is really not very possible in the States. It's most IVF programs in the States cryopreserve anything that's left over. And about 4 or 5 years ago, Greece passed a law that you could only transfer back 3 embryos at any one time. And there is a program at the University of Athens that was started by somebody who worked in my lab a long time ago that has never started a cryopreservation program. The university hospital simply did not want to get involved in these large banks of frozen embryos. So they don't freeze any embryos at all. So they frequently have left over some very nice, normal appearing, and they always do their embryo transfer at the 4-cell stage. So they have nice 4-cell embryos left over, nice 8-cell embryos left over. So I'm not going to go through, we've published a couple of papers we were able to, with two small groups of 8-cell human embryos, we were able to do what's called whole genome microarray analysis, which means that we've been drowning in data for about 2 years. And we were able to compare the microarrays of the human embryos with 2 lines of human embryonic stem cells and with some fibroblasts before and after induced pluripotency. Because as luck would have it, those microarray platforms were the same for those cell lines. We were just lucky that Yamanaka did exactly the same adjuvant microarray that we did. And one of the most interesting things that we found, what makes early embryos divide? How is cell division controlled in the early embryos? Because they're going through cleavage events and each time they cleave, they get smaller. And so what controls that? Because a very common phenomenon in human embryos is something called fragmentation. So this out of this early work that we did, so now we're probably not lined up with that. This is just a picture of a human egg. So now I want to remind a couple of things that sometimes gets overlooked, is that mammalian development, the most overarching fact is conservation of maternal resources. So for instance, as a good example, it's very difficult to keep your mouse pregnant with 1 embryo. 1 embryo doesn't put out enough signal for that mother to think it's pregnant, and you lose it. So there's a lot of pressure on the embryo itself, not just the maternal environment, but the embryo itself, to keep signaling the maternal environment that it's coming along. And the signal must be timely, it must be robust, it must double every day or 2, or that embryo is rejected, because there is not to be any time spent on an embryo that's not appropriate. The other thing to remember is that approximately 80%, I don't think these data have changed, approximately 80% of human eggs fertilize and cleave, but only about 8 or 9% of those eggs develop offspring. So if you go through all of the IVF data, you realize that most of the eggs that fertilize and cleave do not end up in offspring. We know that the timing of the hormone injections is really critical to the success of human IVF. And we know that you can only successfully fertilize a human egg in a relatively short window. It's got a few hours in which you can fertilize it and give a baby. 2 days after it's been ovulated, for instance, it won't. And so this suggests that factors that are essential to fetal development appear late in egg maturation, and they're very short-lived. We don't know what those are. All right? So that was it's always been one of the big questions. What are these oocyte fetal development factors, and what is the role of hormones in their expression? Now this is just a scanning EM example of the kind of 8-cell embryo that we were interested in getting from our Greek colleagues. This is a scanning EM by George Nikas, who is the person who does, the Greek electron microscopist who does this sort of thing. The zona pellucida has been removed, but we wanted a perfectly symmetrical 8-cell embryo to study. And we got 2 pools of 5 of each of these. Now, this is the program that I described to you a little bit earlier. We've had the good fortune to do this. This has me on an airplane to Athens on a regular basis. And this is the microarray. So now we've made this huge database that has our microarray data plus the 2 fibroblast lines before and after induced pluripotency and 2 human ES cell lines. So this is an enormous amount of data that we've been slogging through. Now, the first thing that we were interested in, as I mentioned, is we wanted to look to see cell cycle genes. What's different in the human 8-cell embryo and cell cycle relative to these other 2 cell types? And what about chromosome duplication itself? What about DNA replication, which has been an interest of mine for a long time? I just wanted to remind you quickly of what a cell cycle is. Now, this is a normal somatic cell. The cell cycle begins with some kind of growth factor magic stimulation of some kind of receptor. Early in gap 1, which is the time right after cleavage, cyclin D interacts with its kinase and it begins to phosphorylate RB. And this turns out to be a very important gap 1 checkpoint. Retinoblastoma gene, which was one of the very first cancer genes identified, is thought to be the reason that many cells stay quiet in gap 1, including human adult stem cells. This is thought to be the reason adult stem cells are quiet until called upon. Now, when you get a growth factor, what happens is that cyclin complex phosphorylates retinoblastoma and begins to inactivate it. It inactivates it enough to release some transcription factors. So the reason that retinoblastoma stops the cell cycle here is it physically binds the transcription factors that are important for S phase. So when you begin to inactivate it, you release enough transcription factor to synthesize cyclin E. Cyclin E complexes with its kinase. It then completely inactivates the retinoblastoma gene. And you go on then to release the transcription factors necessary for cyclin A formation. It forms it with its kinase, and now you go on to DNA replication. So now, with that in mind, one of the most interesting things we found in the 8-cell human embryo is that RB is silent. The gene's not expressed. The other interesting checkpoint in G2, We1, is also silent. So this early cleavage phase embryo doesn't seem to have any of the normal checkpoints. Cyclin E, cyclin A, and another oncogene called MYC are way overexpressed. So in this particular system with RB off, these cyclins are very high. And we found, as an aside, that the circadian oscillators seem to be overexpressed. And I won't talk about that today, but we're sort of working on whether or not circadian oscillators are in charge of the cell cycle. So now, just to remind you quickly that embryonic stem cells are programmed to divide relentlessly. Adult stem cells are programmed not to divide. RB is responsible for the G1 arrest in adult stem cells. This is the most commonly dysfunctional gene in almost any cancer. How then does this early embryo maintain fidelity of chromosome replication and duplication to daughter cells? So the lack of RB kind of is consistent with the fact that very early cleavage stage embryos, even in the mouse, don't seem to require any growth factors to do their thing. They just really want to be left alone. The lack of We1 suggests also that the G2 checkpoint is quiet. So how then do you faithfully allocate new chromosomes that you've duplicated to each cell if you don't have any of these checkpoints? So that's the big question. One possibility is that cyclin E and cyclin A are actually controlled by circadian oscillators, and we won't go into that because that point. So we looked at a number of studies in order to develop a whole new list of cell cycle genes that might be important in this. And I actually am not going to take the time to go through these, but we used 3 different studies. We made a long list of the genes that these studies found were involved in cell division. We came up then with 3,803 elements in our gene data set of which only 256 had been identified as cell cycle genes. So we have a lot to learn just about the basic cell cycle. And 24% of the genes that were identified were actually unknown. We don't know what the functions are. So there's a lot to be learned about this. The ones that, whoops. Can you go back? Yeah. So the ones that we found that were overexpressed in the 8-cell stage include the cell cycle regulators I mentioned before, plus some of these. And many of these are involved in chromosome lineup and chromosome checkpoints to make sure that you have the right number of chromosomes in each cell. Now, two interesting genes that may partially explain really commonly observed human embryo defects are the aurora kinase family and this EMI1, which is an inhibitor of anaphase. Inaccurate or inappropriate expression of either one of these genes leads to multiple centrosomes and fragmentation. And these are very overexpressed in the human embryos. How these are controlled is not clear. So if you look at this then, the absence of the cell cycle checkpoints suggests that there's gene duplication may be more important in these early embryos than accurate chromosome allocation. So when I thought about this, I realized maybe it doesn't matter early on if you have exactly the same number of chromosomes in each cell. And this is a very provocative thought because everyone has thought for many years that it's extremely important that you get perfect chromosome allocation early in the embryo or the embryo will fail. But if you look at it, if you go through the numbers, it might be more important to this embryo to make multiple copies of the genes that it needs to signal the mother that it's developing. So what if the most important thing this embryo has to do in the first 24 to 48 hours is begin to make multiple copies of chorionic gonadotropin gene, for instance? And maybe that's more important to its survival than making sure it gets exactly the right number of chromosomes in each cell. Now, so if you look at the zygote, so then you come to the question, well, if you're just going to do this willy-nilly, what is the minimal number of cells that need to be perfect for you to end up with an embryo, okay? And so this is just a schematic of early embryo development. You've just seen these elegant pictures from Janice. So you can see now that, as she described, at about the 8-cell stage, between the 8-cell stage and the 16-cell stage, if you put a bunch of styrofoam balls together, you end up with one or two trapped in the middle. And that's about what happens somewhere between the 8-cell stage and the 16-cell stage. And I've indicated that here as these little yellow balls. Those are going to give rise to the inner cell mass by all of the elegant mechanisms that Janice just went through. So in fact, those are the only two that have to be perfect. Right? It isn't probably quite so important that the trophoblast is maybe a little bit aneuploid. So if you take that further, in this model, that means that the first cleavage probably does have to be perfect. So how that happens isn't clear, but the first cleavage has to be perfect. But the next cleavage, only one has to be perfect. The cleavage after that, only one has to be perfect. And the cleavage after that, only one really has to be perfect. And then you can still give rise, as long as they're going to be the inner cell mass, to the perfect inner cell mass. Now, how often does that happen? Now, I want to point out that this is just a theory, but I think this is very provocative because a number of studies actually support it. So there have been a few studies of deriving stem cell lines from individual cells of embryos. And the success rate of this is quite low. And it could be because it's difficult to do, these cells need some more support. Or it could be a reflection of the fact that they might be aneuploid. So this was one study that was published several years ago now, in 2007. And they tried to make a line of stem cells from individual cells from 2-cell embryos. And they only came up with 4 stem cell lines. Then they tried to make stem cell lines from individual cells of 4-cell embryos, and they only came up with 1. Then they tried to make them of 8-cell embryos, and so each time the efficiency of these early stages was very low. But the efficiency of deriving stem cells from the blastocyst stage when you already have an inner cell mass that's now multiplied many times over, was about, what does it say there, 18%, 80%. So it's a very high efficiency once you get to the inner cell mass stage, and a very low efficiency when you're trying the individual blastomeres. Now that would fit the idea that perhaps many of those blastomeres are aneuploid and they simply don't continue to divide. There's other explanations for that too. Then the same thing happened from stem cell lines which tried to be derived from individual human embryos. They only got 2 stem cell lines from 91 blastomeres that they tried. Then there was an interesting study a couple of years ago of stem cell lines derived from 4 cell stage embryos, where they used 2 4-cell stage human embryos. That gave them 16 individual blastomeres, and they were only able to derive 2 ES lines, only one of which was normal. Then there have been a number of studies. There's a new way of screening embryos in fertility clinics that's actually fairly controversial. And one of the ideas is that you would take a cell from like an 8-cell embryo, analyze that one cell, and make a decision about whether or not that embryo is normal, and you should transfer it back to the mother. Now this is quite controversial because there are some clinics that think this is the wrong thing to do, other clinics which think that's an appropriate thing to do. And there's a big argument over how best to do it. So one of the methods that it's used is this method called FISH, which is an immunofluorescence method that identifies each chromosome individually. And the other is a more microgenetic approach where you're looking at single nucleotide polymorphisms to try to characterize the cells at a genetic level. And when you look at the bottom line from these 2 studies from one large laboratory in New Jersey, is that when you look at embryos early on, and then you look at them later, you discover that early, many more of the embryos appear aneuploid and defective than they do later. So that's known. If you now make a decision about an 8-cell embryo, when you get to the blastocyst stage, what you might discover is that although it was abnormal, the one blastomere that you collected at the 8 to 16 cell stage, the 4 to 8 cell stage, was abnormal. By the time that embryo gets to the blastocyst stage, the inner cell mass cells are normal. So this is a controversial area, but it kind of supports this rather provocative notion, and this is a human blastocyst hatching. So what we're looking at is, this is the trophectoderm that Janet so beautifully described. This is the inner cell mass, probably just with maybe at the beginning of endoderm here. This is a hatching blastocyst. So one of the take-home messages is it possible that early on, the only cells that you really need to be perfectly euploid, perfectly chromosomally normal, are the cells that are going to give rise to the inner cell mass, and that many of the other cells could be aneuploid and still form perfectly functioning trophoblast. That's just an idea that I think it's interesting to throw out. I'm going to end this talk with, I think I've got a slide here. Yeah, this is a little bit about the Bedford Foundation. We have a wonderful board of trustees that spends a lot of time raising money for us. We have an ethics advisory board that tries to keep us out of jail. And then this is just a list of the scientists that have contributed to this work through the years. And I'd be happy to answer any questions, although we're running a little behind now, and we definitely don't want to lose out on any of the food that's coming next. Laura?
So do you think then that it's those cells that manage to maintain their normal chromosome complement that become by default the inner cell mass?
That's going to be, Dr. Rosan's going to tell us that. Are those the ones that have the right polarity that get in there? Or is it random? Is that
Right.
why only 8% of
Right.
embryos,
That was
is it a
the
random
alternative,
event early on? I don't think we know that. Yes?
Hi, you mentioned day 3 biopsy of human embryos, maybe giving higher rates of aneuploidy. What is your opinion of trophectoderm biopsy, which has been done a little more frequently nowadays?
I think
We're just
these,
looking at
I think
trophectoderm
that,
cells.
I'm not in this field anymore, but I think the idea of deciding based on any kind of chromosome analysis what an embryo is going to do is tricky. I think if you're going to biopsy that embryo because you're looking for a specific genetic defect that you know runs in that family, and you're trying to screen for a specific genetic defect, that's probably reasonable. But to try, I don't think that cell biopsy is going to give us a really good indicator of what embryos to put back. Maybe Dr. Rassant doesn't agree with that, but yeah, she agrees. I think that that's going to be, the chromosome analysis at this very early stage is going to be flawed for some time. Ken?
Do you see RB or any of the checkpoint genes coming back on in late pre-implantation development? And if so, is it specific to inner cell mass?
We have microarray data on the blastocyst, and it's just sitting on the computer. Jaime?
Is there evidence of aneuploidy in other primates? Let's say chimps, or has any look?
Yes, that has been done. I'm not up on that, but yes, there is evidence of aneuploidy in primates. As a matter of fact, there's some very nice papers by Carol Ziomek, I think, looking at both aneuploidy and I think it's pretty common. David, do you have any thoughts on that?
Yeah, I definitely. Carol Brenner has shown, in working with a group out in down in Tulane, down in the New Orleans University. I have a question about these rapid cell cycles and what's going on early. Then there's been a very interesting series of papers coming out of the Cole Laboratory at NIH that has shown at the 2-cell stage to 4-cell stage expression of Z-scan4, which came out of looking at expression, differential expression, I think in mouse, but other species seem to show something similar, which promotes recombination. And interestingly, we had published in Nature Cell Biology a couple of years ago showing telomere recombination, again, during this pre-implantation stage of development. And the group from Louvain has shown an enormous number of copy number variants in reputedly normal embryos. So my question is, could pre-implantation embryo development also be like meiotic recombination, a phase of development when recombination is again
still happening?
More diversity genetically, and could this high rate of aneuploidy be just kind of the fallout, the downside of this genomic instability, which may have some adaptive advantage?
Do you have any thoughts on that?
Sounds
like
like
an
a
interest-
discussion.
All right, one more question. Terry?
I was curious, it's actually more theoretical than looking at the science here, in terms of the approach of how we study things. One way to study is looking at the hows, which is what we heard first from Dr. Son. And that's what a lot of us are interested in, is how do things happen from one place to another. Your approach is a little different, and it's looking at the what. And I find your approach sort of stepping back, it's a more Darwinian approach. In a way, if you think about it, as to what is going to be successful.
Yeah.
So I think it's sometimes useful to understand. Sometimes we get caught up in one model, and we don't get to looking at the more macro picture that you're doing. And I think you need to have some sort of combination of the two going on at the same time. Because sometimes we don't step back and look at the picture that you're presenting. So I just want to offer that as an observation.
You want to help us slog through some of these genes? You're drowning in genes.
No.
Thank you very much, and I think it's time for our little bit of our break, and then we're going to come back and listen to some great calcium fluxes.